Picture this. You're a lone cell at a crowded street market. On your own, you can barely see past the next vendor's stall. But pack thousands of you shoulder to shoulder, and almost magically, you start sensing what's happening half a block away. That, in a nutshell, is the supersense scientists have just uncovered. Cells can feel mechanical changes up to 10 cell lengths farther than we ever thought possible, and that changes how we think about wound healing and how cancers spread. First, let's talk about the good side of this talent. In healthy tissue, collective sensing is like a well-choreographed dance. Skin cells close a cut in synchronized waves. Gut lining renews itself without leaving gaps. Embryos sculpt organs with millimeter precision, all because neighboring cells share mechanical gossip. Who's pushing? Who's slacking? Where's the soft spot we need to fill? Now, here's the twist. Inside a tumor, the very same talent becomes risky. When cancer cells pack together, they don't just react to their surroundings, they remodel them. Imagine a group of hikers laying down planks to cross a swamp. Each step firms the ground and hints at the next direction. Tumors pull collagen fibers tight, distort nearby ducts, and carve out new roads of softness and stiffness. Those roads whisper to clusters deep inside the mass. Psst, escape route, 300 microns east. And some clusters listen. The key point is this. A cancer cell's fate isn't dictated by genes alone. How tug-resistant the neighborhood feels can flip genetic switches, change how sticky a cell is, even decide whether it settles down or keeps roaming. That's why drug trials now target force-sensing hubs, and why bioengineers build tiny microterrains in the lab to see just how far this long-range sensing can be stretched, or shut off. So how do cells pull off this long-view trick? They invest in hardware. Picture protein cables inside each cell, clutch-like grips to the matrix outside, and zipper-tight junctions that weld neighbors together. A shove in the front row travels through those connections, gets amplified, and arrives as a quick message in the back. Suddenly, the whole sheet knows which direction is stiffer, which is softer, and who should lead. Two big consequences pop out. 1. Collective wisdom. Rear cells agree to follow leaders they can't even see because the mechanical signal arriving at their own feet already encodes the landscape ahead. 2. Landscape rewrite. Every probe leaves a footprint. Fibers realign, crosslinks snap or form, and stiffness becomes history-dependent. The second time a cluster passes through, the road is already paved. Engineered lab substrates make this crystal clear. Tweak the spacing between adhesive dots or change how quickly stiffness ramps up, and a smooth, surfing wave of cells turns into a jittery, stop-and-go traffic jam. Dampen a force processing hub like focal adhesion kinase, and suddenly the same terrain becomes hard to read. Cells still grip, but they lose their long-range intuition. Because mechanics and biochemistry never stop talking, muting that intuition can ripple outward. Gene expression shifts, inflammation signals change, immune patrols wander off course. It raises a tough question. If we shut down harmful migration, do we risk hampering helpful repair that runs on the same wiring? Think about city traffic for a second. One red light tells you nothing, but watch thousands of cars and patterns jump out. Rush hour waves, hidden shortcuts, bottlenecks locals know by heart. Researchers see similar rules in collective mechanosensing. Stamp tiny islands of softer or stiffer gel into a dish, and big clusters self-organize into streams, branching roadways that funnel force along preferred corridors. Those corridors guide where growth factors pool and where microgaps open in vessel-like layers, prime exit ramps for cancer cells. Here's where it gets exciting. Engineers are now printing stiffness roads on purpose. They test which cell types choose which routes, and whether blocking a few strategic junctions can reroute invasive cells away from vulnerable organs while still letting everyday repair happen in skin or gut. All of this hints at a future design goal called measured mechanical privacy. Imagine shielding fragile tissues from long-range cues that lure cancer cells, while keeping the short-range whispers that heal a paper cut. Clinically, that might look like injectable gels that blur stiffness gradients around a suspicious lesion. In the lab, it means mapping how each cell type balances distant and local cues to explain why some organs are metastasis magnets while others act like cul-de-sacs where motion stalls. So, where does this leave cancer research? We can't just ask, what mutations drive escape? We also have to ask, what does the escaping cluster actually feel? Future trials may pair gene-targeted drugs with materials that damp or redirect force signals, like acoustic panels in a concert hall, quieting the echoes that beckon cells toward distant organs. Let that sink in for a second. Recap and takeaways. So here's what we covered today. 1. Cells aren't lone rangers. In a crowd, they sense mechanical changes up to 10 cell lunxers away, farther than we ever expected. 2. That supersense keeps wounds healing and organs growing, but in tumors it can carve out escape routes. 3. The secret sauce is a physical network, protein cables, matrix grips, neighbor junctions, that turns tiny tugs into tissue-wide intelligence. 4. Tweak the mechanics and you tweak behavior. Drugs, engineered gels, even subtle stiffness gradients can amplify or mute long-range cues. 5. The next frontier is mechanical privacy, protecting healthy tissue while denying cancer cells the whispers that help them roam. Here's your personal challenge for the week. Ask yourself, what distant influences, stress, posture, daily movement, are quietly shaping my own tissue mechanics? Pick one doable change, maybe a lunchtime walk, a strength training session, or a posture check, to give your cells a healthier physical signal. And finally, build an anti-cancer internal environment. Aim for nutrient-dense meals, quality sleep, and moments of calm. Small mechanical and metabolic tweaks add up. Remember, your cells are listening to more than chemicals. They're tuned into the forces around them. Give them the right cues, and they'll work for you, not against you. Thanks for hanging out with me today. If you found this helpful, share it with a friend who geeks out on the hidden lives of cells. I'll catch you next time. Stay curious and stay kind to your tissues.